Today we're going to talk about cytogenetics. As you can see, I've got some definitions on the board right now. We're going to talk about cytogenetics. We're going to talk about how they use quinacrine as a dye substance, and we're going to talk about Giemsa, and we're also going to talk about a method called chromosome painting. So as you can see right here, I've got a karyotype displayed for you, and all a karyotype is, it's an image that displays the individual's chromosomes. So if we have a newborn in the hospital and the parents wanted to figure out the karyotype, well, we can do some cytological analysis in order to figure out whether this individual is regular, is normal, or if this individual has Down syndrome, which we would be able to distinguish based on how many chromosomes they have on chromosome 21, if they had a trisomy, if they had a third chromosome. So what we're going to do is we're going to go through each of these definite definitions, and we're going to answer what each definition means, so you guys can learn along this process. Okay, so if someone was to ask you what is cytogenetics, you would answer, it is the study of stained chromosomes. Okay, these are people that work in the laboratory in order to give you predictions as to whether your child is going to have Turner syndrome, if your child is going to have Klinefelter's, if your child's going to have trisomies, or if your child's going to be polyploid in any way. So these are cytogenetics, and geneticists as well, but cytogenetics use stained chromosomes to figure these abnormalities out. And one of the ways they do so is by using different equipments, right? If you're going to be working with the field of study, you need to have some stuff that you're working with. Well, quinacrine is a dye that they use. And actually, let me change the color here. Quinacrine is they call it a fluorescent dye, because when you introduce ultraviolet lights with the quinacrine compound, what happens is the substance itself gets excited, and that's what produces the stain on the chromosomes. So you're able to depict the chromosomes themselves, and you can distinguish whether this is chromosome 5 or whether it's chromosome 8. So these are dyes that they use in a laboratory. And then you have something else called Giemsa, and this is another dye that they use, although this is a non-fluorescent dye. And we're going to talk about the differences between what it means with the fluorescent compounds and what are non-fluorescent compounds. But for now, what we need to understand is that both of these are dyes used in the laboratory in order to detect chromosomes that might be malfunctioning in some way, or aneuploid. And then the most commonly used method in the laboratory by cytogeneticists is a technique called chromosome painting. And essentially, this is what involves this karyotype right here is produced as a result of using this chromosome painting technique. This is how you get this karyotype right here. And let me just label that this is a karyotype, because it's an important term, karyotype. Okay? And you get this karyotype by doing this chromosome painting, and which is technically using DNA segments in order to reveal the chromosomes themselves. And I'm going to explain this in a second. So people are probably wondering right now, I understand what cytogeneticists do. They study stained chromosomes in order to tell me if my child has Down syndrome or if my child has this disease. And I also understand that they use quinacrine and Chiamza to do that in order for the chromosomes to be stained so you can see them. And by the way, they do this in dividing cells in mitosis. That's really important. They do this using cells that are undergoing cellular division in mitosis. So, you're thinking, I understand that they use these dyes in the lab so that they can stain the chromosome so they can view it under a microscope. But what is chromosome painting, and why is it the most famous, more commonly used method in the laboratory? Well, let's talk about chromosome painting and why and how is it that chromosome painting can give us this image right here of a karyotype. Well, in chromosome painting, like I said right here, you've got these segments of DNA, and these segments are often called probes. So you've got these little segments of DNA that have already been pre-cut in the laboratory. Remember, these have been ready. These are ready to go. They've had these ready for a few days now. All they have to do is inject them within a solution in which they're trying to figure out how many chromosomes are present. So you've got all these little segments of DNA, and what they do is now, let's say you've got chromosomes right here in a solution. So these are chromosomes right here. And so what they're going to do is they're going to inject, okay? They're going to inject because in a solution, you're not able to see the chromosomes. The chromosomes are everywhere. You're not able to distinguish whether this is chromosome 1, if it's chromosome 5, because chromosome 1 is the largest chromosome, okay? So they decide which chromosome it is based on the size, the length, based on the position of the centromere, okay? So in order to do that, you have to be positive. So they inject these DNA segments, also called probes, and let me just label that for you. These DNA segments are also called probes. So let us write that down right here, probes. And they will inject this into a solution that contains chromosomes. So once you do that, the result that you're going to get is you're going to have these DNA segments become attached to the chromosomes. So something like this. Okay, this is going to happen, and once that happens, you can then tell the chromosomes themselves. You can be able to see, oh, okay, well, this DNA segment is attached to a substance, and based on that substance can only be a chromosome because of the particular DNA synthesis needed for this reaction to occur.